So this is you saw how complicated it was. This is very simple. You have you have here on the outside, this side out here is the outside of the mitochondria, but inside the cell. There's your cell membrane, correct? Why is this? Here's your cell membrane. This is the cytoplasm. Remember that the cytoplasm is the liquid part of the cell plus the cytoskeleton. All this stuff, that's all those little vesicles and everything, that's called the cytoplasm, the kind of fluid part of the cell. Okay? Is that okay? Here in the cytoplasm, this is what's happening. You need to know this. Glycolysis happens in the cytoplasm. This is what you need to know. You don't need to know all that crazy stuff I was just showing. The AP kids need to know that. They're stressing about it right now. Glycolysis happens where? In the cytoplasm. Glycolysis does not need let me write it really quick, small, does not need oxygen gas. No oxygen necessary for glycolysis. And so because of that, we call it anaerobic. Yeah. Yeah, it's anaerobic. It's spelled in a packet, Raven just told me, so we're good. So anaerobic means no oxygen is necessary. That's what this means, all right? So rather than putting an extra little dash there, I'll just say that's what this means, and I'll put it in parentheses. Anaerobic means it does not need oxygen. Glycolysis does not need oxygen. Glycolysis is taking glycolysis. Glyco comes from glucose, so sugar. What does lysis mean? We must have said it like 100 times. It's one of those vocabulary words from first semester again. Means break down. When you lyse something, you break it. When a cell lyses, because it's in hypotonic solution, it breaks apart. Lysis means rupture or break. So glucose has 6 carbons. You break it into 2 what? 2 what? 6 carbons gets broken down into 2 what? 3 carbon molecules. That's all there is to it. Again, glycolysis is all about taking this 6 carbon molecule called glucose and breaking it down to 3 carbon molecules. 6 divided by 2 is 3. We call that glucose, that 3 carbon molecule, pyruvate. You need to know the word pyruvate probably. But in any case, I'm not sure they're going to ask you for it on your final for the year, but I would know it. So this 1 6 carbon molecule, every one of these glucoses that you eat from your sugar or from your candy bar, from your bread or whatever, the glucose is out here in the blood. It comes in through the channel, and that 6 carbon glucose gets broken down into 3 carbon molecules called pyruvate. That's all. In the process, while that's happening, by way of breaking that down, we end up making let's be clear about this. I want to be honest with you, even though it might, I hope it doesn't cause you any trouble. But I'm going to say this. I'm going to go ahead and put a little a little footnote over here. You do make a net. You make a net. Everything in green is what I'm making. A net of 2. All right. However, what happens is you use 2, as you just saw, ATPs, and you make 4 ATPs. So that means you're making a net of, remember the word net, of 2 ATPs. So you use money to make money. You use 2 ATPs to make 4. It's a good deal. The really, really important part, though, that's it. That's useful. It is. That's very useful. Except that the only problem is that that 2 ATP is not enough for you to live on. If that's all you did, if you were a yeast cell, that would be fine. A bacterial cell sitting in a, in a solution, okay. But you're a human being, you have a lot of energy needs. You need a lot. You have a lot of energy needs, and 2 is not enough. There's still more energy in this pyruvate. You did get a lot of energy out, but you didn't make ATP with it yet. And this is an electron. This is what we call an electron carrier. Again, another footnote here. Let's use a different color. This is known as an electron carrier. NAD, just another molecule. I'm not going to draw it. You don't have to know its shape. Just know NAD is called an electron. In fact, let me just give it a title first and then put what it is. An electron carrier. That's all it does. It carries electrons. So NAD, it's nicotinamide. I forget the rest of it. NAD plus, so it can take an electron. You add an electron with a hydrogen, and you get a hydrogen ion, and you get NADH. If you had a hydrogen ion, you add an electron. So now this molecule has an electron in it. NADH. Are we all clear on that? So NADH has an electron. NAD plus does not. So now you have your energy. Where's your energy? You got energy here that you pulled out of glycolysis. You invested 2 and you made 4 and you got 2 net. You made NADH. That's good. That's going to go to, this is going to the electron transport chain. You'll know it as the ETC. They'll show it sometimes as ETC, electron transport chain. This here is using an enzyme. Glycolysis is anaerobic, right? Does not use oxygen. It uses an enzyme to phosphorylate. Let me just, instead of, to add phosphate to ADP to make ATP. And that's what it's doing. This process can only do it can only make 1 ATP at a time. I had a student that just knitted a hat, and it was really beautiful. You've seen her. I don't know if you know her. She just knitted a hat. She crocheted a hat. Really beautiful hat. Don't put things away, especially if you don't know vocabulary words for your quiz. So she just crocheted a hat. Her idea was she could sell them. She could crochet a hat and sell them and make money. Do you think she could do that?
Yeah.
It's a beautiful hat. Well, you don't see a problem with crocheting a hat and trying to make money that way? Okay, it takes her 6 hours to crochet a hat.
I mean, that's a lot of time, but I don't
How much could you sell the hat for?
probably like $4
Huh? $4 or $5, the crocheted hat, for 6 hours. So you're going to have a job. Hold on, I'm not done. Listen to what I'm saying to this so I can make my point and you can leave. If you sell that hat for $5, let's say $10, and it takes you 6 hours to make it, how much money are you making every hour?
Like,
No, if you sell it
like,
less than minimum wage, that's not enough to live on. So this is the problem with pay attention before you leave. This is the problem with glycolysis. It's only making 2 at a time. It would take, it's not making enough money for you to live on. Well, tomorrow we're going to see how we make the real bucks. All right, have a good one. So luckily for us, if there's oxygen, that NADH that's carrying those electrons can take that in here. That pyruvate can go in here, and we can squeeze out more ATP out of this. At this point, this is, let me write over here on the side, as I tell you should write in Cornell notes. Glycolysis that's happening in the cytoplasm, that's not using oxygen, is doing substrate level phosphorylation. What that means is you're taking ADP and turning it into ATP. Phosphorylation means you're adding a phosphate. Adenosine diphosphate turns into triphosphate. All right. This is good because this is our cash right here, our greenback. This is what we want. But it's happening only one at a time using an enzyme, hence the word substrate, because enzyme needs a substrate. So it's doing one at a time. And it's very slow. It's slow, produces very little ATP per glucose. In fact, it produces a net of 2 per glucose, 2 ATP. So it's very slow, not very good, but it's okay. It's good enough for yeast. It's good enough for bacteria. Now, there's 2 choices. So we're going to do a flow chart right now. So a decision chart. 2 choices to sell. The choice is not a decision as much as it is something that is determined by the resources that are available. If there's oxygen, if O2 is present, if there's oxygen gas, if you're breathing, then the pyruvate and the electron, the pyruvate's carrying the carbon and some electrons, and the electron carrier known as NADH go into the mitochondria. If there's oxygen present. If there's no oxygen present, if no oxygen is present, then it goes to something called fermentation in the cytoplasm. It stays in the cytoplasm, and pyruvate goes to fermentation. Are we clear on this? So there's two kinds of fermentation. Two kinds. There is that fermentation that happens in humans and other mammals. In fact, most vertebrates, in fact, as far as I know, all vertebrates, let me just put vertebrates. Do you know what the word vertebrate means? Yes? Things with spines? Most animals, let's just say animals. Forget it. Let's just say animals. You are an animal. You know that, right? In animals, we do something called we make something called lactic acid. No CO2 is made. No CO2. But we do use those high energy electrons. We use the NADH. We use the NADH. So NADH gets turned into NAD plus. And we make lactic acid. Now, this is an acid. If it builds up too much, it hurts you. Not good. But the reason we do fermentation in animals, and you and I do it, is because there's no oxygen, so we're going up 8 flights of stairs. We're breathing heavy, okay? We're running our laps around for basketball, up and down the stairs. I see you sweating in the hallways and running around the laps on the track and out of breath, your chest going up and down. I can't breathe. This is too. I see you. Yeah. That means you're running out of oxygen. You're building up lactic acid. Now, some people say that that's why muscles hurt. It's not. Muscles hurt because you tear some. You do slight damage to it as you exercise them. But lactic acid can be deadly. Don't put anything away for a minute. Wait. The reason you do it is so that you can get NADH, so it can come back to glycolysis and take up another electron, so you can keep making 2 ATPs at a time. Because if you don't do it, you're going to run out of NADH, and that means the whole chain stops. The whole pathway stops, ladies. So you have to keep doing it. The interesting part, and this is where I've seen test questions come up, where people lose it, okay? This is where some people lose it. Lose the script, as it were. And the interesting part is that in yeast and some bacteria, yeast are eukaryotes, they don't make lactic acid, they make alcohol. And they do produce CO2. They produce a gas. They don't make lactic acid. They make alcohol and they produce a gas. We don't produce CO2 when we do fermentation. Yeast do. That's how we use yeast to bake bread. When you put yeast in dough and put it in the oven, they eat the sugar. They do fermentation. Obviously, inside the dough, there's no oxygen. They make CO2 and it puffs out those little bubbles, right? And as the bread bakes, and you cut some bread, you see all those holes, that's where the yeast were growing. The yeast are all dead, they got cooked. Also, that's what happens when we make alcohol. When you make champagne, where do the bubbles come from? CO2. The yeast eat the sugar, make alcohol, hence alcoholic, right? That's what champagne is, alcohol. But it also has carbonation because of the CO2 gas that's being made. This idea that animals don't produce CO2 during fermentation and yeast do, that yeast make alcohol and animals make lactic acid, is one of those key points you have to understand. Only when there's no oxygen present. All right, we'll finish this tomorrow. And this is testable for both AP and honors. I've seen questions on the OST as well as the AP exam. The difference between the two, I've seen questions where they asked you, they've asked you, what's the difference between yeast fermentation and animal fermentation? 2 key points. CO2 gas is produced in yeast. And the fact that you produce alcohol in yeast and lactic acid in animals. 2 big differences. In both, you're replenishing your NAD+, so that you can go back and pick up another electron. But the key difference is you're going to produce CO2. So if fermentation was happening in an animal cell of some kind versus a yeast cell of some kind in a test tube, and you took these cells and you put them under the right conditions so there's very little oxygen, what you'd expect in the yeast cells is bubbles. And what's in those bubbles? CO2. Now those are the kinds of questions they're going to ask. Like, what is in the gas? When a scientist measures, they'll give you, they'll put this in an inverted test tube, they'll fill it with water, and there'll be gas. And as the cells continue to do their bubbling, this gas will push in and expand, and it will push the water down because of the pressure from the gas being produced. They'll ask you, what is that gas? CO2, because that's what animals and yeast produce. They produce CO2. They don't produce oxygen. They use oxygen. So it really comes down to, do you understand that in cellular respiration, you're using oxygen, you're producing CO2. But if they tell you that they put an animal cell in that same experiment, then there is no gas. I've seen some simple questions like, okay, there's some bubbles, and then you have, so they have simple questions like, for instance, you have 2 of these test tubes. You have A and you have B. Except that one of the test tubes has no bubbles. One of the test tubes, the cell's there, same conditions, everything's the same, but there's no bubbles, so there's no space. The test tube is completely filled. So a real simple question is A or B, which one's an animal cell, which one's a yeast cell? If there's no oxygen present with glucose, which one is yeast cell? What would be yeast here and animal here? Why? Because animal cells do lactic acid, where CO2 is produced by yeast. So that's the kind of thing that they like to ask. They're not going to They might ask you a multiple choice question, which is which of these is something that that a yeast would produce in anaerobic conditions. Choose all that apply, and so they make it a little more difficult with choose all that apply. They would put alcohol, CO2, oxygen, NADH, NAD plus, lactic acid. They could give you a whole list, and obviously yeast are going to make alcohol and CO2. They're going to use up glucose. Where animals are going to make lactic acid, use up glucose, produce no CO2, they're all going to use NADH and produce NAD+. They're not going to make NADH. NADH is made in glycolysis. It's another trick question, isn't it? Can easily mix that up. Could you close that computer, pay attention, take notes? So NADH is made in glycolysis and used in fermentation. So fermentation is kind of just a short stopgap measure. Stopgap measure, meaning it's something we do and yeast do when we have no oxygen, just so we can survive. That's all there is to that. So let's again just to keep touching on what we've covered already. We said glycolysis. Check. Happens in cytoplasm. It's anaerobic. No oxygen being used. Multiple steps. Several enzymes. We'll talk about cellular regulation tomorrow. Or its regulation tomorrow. It's making NADH. It's using NAD plus. It's using ADP. It's making ATP. 2 net, 4 total. It's using 2 to make 4. It makes pyruvate, a 3-carbon molecule, with some intermediate steps in between. Again, the regulatory steps we'll talk about. We talked about fermentation. There's not really a lot more to talk about fermentation. It's pretty easy. It's the easiest of the energy pathways. So that's 1, that's 2. Fermentation is not cellular respiration. Is that clear? Are we clear on that? Cellular respiration is not. Instead of 2, let's draw this, because one of the questions in here is what are the steps to cellular respiration? That is, glycolysis is one of the steps. And 2, another step to cellular respiration is something called the Krebs cycle. Not Krebs. Now, it's Krebs cycle, capital K, because it's a guy's name. The other thing is, it's also known as the citric acid cycle. And it's also known as, I always forget one of the 3. Always a different third as well. Also a different 3. Krebs cycle, citric acid cycle, tricarboxylic acid cycle. Why do they have 3 names for the same thing? I don't know. Which one are they going to use on a test? I don't know. But the three of them are there. They're all the same pretty much. So the overall, again, this is just overall. What happens is when there is oxygen present, that's the other option, right? Remember, we looked at that fork in the decision-making pathway, and again, it's not a decision. The cell's not like, which way should I go? No, it's just simple. If there's if there's oxygen, then everything's going to go towards the mitochondria, period, done. In a eukaryote, obviously, prokaryotes don't have mitochondria. I keep saying these repeatedly so hopefully it sinks in. Okay, so as the NADH goes right into here, into oxidative phosphorylation. By the way, oxidative phosphorylation is also known as the electron transport chain. Electron transport chain. What do you think you're using in oxidative phosphorylation? Oxygen. So as pyruvate goes into the mitochondria, NADH goes over here to the electron. Why? Because NADH is a carrier. What is the NADH carrying? Electrons. Where is it going to take it? To the electron transport chain. I have only one word to say about this, and I hope you understand and see how clear this is. Duh. Right? Where is the electron transporter taking the electron? To the electron transport chain. That's just like the old joke, who's buried in Grant's tomb? Well, Grant. It's not hard. All right. So here you have this pyruvate, again, a 3-carbon molecule that goes into the mitochondria before it gets to the Krebs cycle. Before it gets to the first step. Remember, pyruvate's 3 carbons, has 3 carbons. Where do those 3 carbons come from? Another good question they love to ask. Where does the carbon come from? From glucose. So the 3 carbons that are in pyruvate come in. First thing that happens is a reaction that removes one of those carbons, and out goes carbon dioxide. Carbon with an oxygen. No oxygen. Your oxygen you breathe in is not used to make CO2 here. Are we clear? They love that question. I've seen it even on the MCAT. Oxygen that you breathe in does not go to make CO2. It goes to make water. This CO2 comes right from, all that CO2 comes right out of sugar. Out it goes. So that's literally the mass that you've eaten, you're breathing out. It's done. 2 acetyl-CoAs are made. Why 2? Because there's 2 pyruvates. Everything's doubled because you took that 6 carbon and you made it 2 3 carbons. So you said 2 CO2s here. It's only actually 1 CO2 per pyruvate. Are we clear? All right. So that's really kind of tricky when they do these summaries. It gets a little bit like you got to think about it for a second. This is less that low-level depth of knowledge question, memorization, and a little more active brain, maybe level 2 depth of knowledge, level 3, where you're just like, okay, maybe I got to figure this out a little bit. So acetyl-CoA is a precursor. The CoA will get recycled. But those 2 carbons that are left from the pyruvate, they're going to leave. And we'll look more in detail in a minute. And the little miniature group of people that met yesterday, you're going to hear some repeats. But I think that's good anyways, so you get to review a little bit. Maybe you'll be answering in class rather than me doing all the talking. So Krebs cycle it's a cycle. It goes round and round. It reuses almost everything. The only thing it's not going to reuse is the CO2 because the carbon leaves. Where'd that carbon come from? Glucose. 4 leave, 2 more per pyruvate. So 4 plus 2 is 6. The original 6 carbons that came out of glucose. They're left. You breathe it out. Done. They're the bubbles in the test tube for yeast. They're the gas in your exhale. And of course, you get 6 NADHs, FADH2, and it's FAD plus, and then it takes on 2 electrons. We'll go then to where? These are electron carriers. Both of these are electron carriers. Where are they going to take the electrons? Electron transport chain. Again, very simple. They're going to take them to the electron transport chain. Boom. We're going to look at the electron transport. Say what now? We're going to go over, we're going to review in a second what is oxidation reduction. They call them redox reactions for a reason. But does everybody get what we just looked at right here? We love to call cellular respiration this. This is cellular respiration as a generalized formula, but there are many, many chemical reactions that are happening in this big overall formula. All right, let's look at oxidation reduction. I'm going to make it as simple as I can. If I'm going to use variables, if you don't mind. It doesn't really matter what variable you use. Let's use A for one variable and B. So these are my reactants. What normally happens, we like to couple, what happens in biology is we couple, a life couples reactions. Every time you couple reaction, you pass on some matter, you pass on some energy, you lose a lot of energy in the form of heat, but some energy gets transferred over to the coupled reaction. So what happens is an A becomes B. A becomes B. Often what happens is it passes off the electron. It gives the electron, so then C becomes D. D, when that electron gets transferred, now you could talk about it as oxygen, right? Because in fact, in cellular respiration, we say oxygen is the last electron acceptor, so that's why it is oxidative phosphorylation. But what happens when you oxidate something, a simpler, really simple, and those of you they're going to go into organic chemistry, those of you whose parents tutor organic chemistry and teach organic chemistry at Cleveland State, right? And make sure your mom gets a copy of this. Well, a simple way of looking at it is that when something becomes oxidated, something becomes oxidated, ED, it's been oxidated. Its charge, the charge on that species, I'm going to call it a species in a chemical term, not a biological term. So it's a molecule or an atom or something. The charge goes up. It becomes more positive. Right. Electrons are removed. You cannot have oxidation without reduction. That's why we call them redox reactions. Oxidation, reduction. Redox reactions. You cannot have one thing being oxidized without something else being reduced. Something has to take the electron if something's giving it. Does that make sense? Okay. So if this one gets transferred over to here, right, then what happened to A? A was what? Was A oxidized or reduced? A gave up an electron. It was oxidized. It became oxidated. They became more positive. More positive. So it is oxidated. Now C became more what? Reduced because it became more negative. So this is the reduced form of that substance. In many cases, oxygen is doing is involved in the process. So that's where we've, in fact, in the first oxidator that that I know of that was known was oxygen, and we see it every day, especially now in the winter, and iron that forms iron oxide, and that's rust. And you can, but when you're talking about oxygen gas becoming iron oxide, Fe is +2, each oxygen is a -1. The Fe became what? What did Fe become? Less positive, so it became what? Reduced, right?
It's less positive. Then it's, yeah.
And then you added oxygen to it, right? So we call it. So the things that, in any case, the bottom line is, guys, when we're talking about this, think of them as electrons. And if they're gaining an electron, the charge is coming down, so it is being reduced, and the thing that gave it the electron is being oxidized. I'm sorry, that just reduced. That's true. Yeah, I said that right.
The one that gets it.
Say what? So oxygen comes in the end and takes the electron. That makes that species more positive, and oxygen becomes reduced because it took the electron. And so you take it and you add, when you're reducing, often another aspect of this, especially in biological systems, when you see at the end that you have a bunch of these, you'll see it when we look at the electron transport chain in just a minute. When we look and we see these H pluses sitting around, it's acidic. That's what H plus means, right? Hydrogen ions means it's acidic. So you have all these H pluses sitting around, and you'll see why in a minute. Oxygen gas comes in, it takes the electron. Oxygen gas is two oxygens. It comes in, and it has to take that last electron. Could someone close that door? It takes that electron and that hydrogen and a couple of those hydrogens. It's not going to take one. It's going to take one electron apiece and, two electrons apiece, because each oxygen has a -2 charge, correct? It's oxidative state. You heard of the term oxidative state from the periodic table? Oxygen needs two electrons. Is that correct? No. Am I talking to myself? Am I wasting? So oxygen needs 2 electrons. It comes in, each oxygen is going to take 2 electrons. So how many electrons is oxygen gas going to take? 4. So it takes 4 electrons, 2 per oxygen. So now it has, here's this, now we have this oxygen atom with a -2 charge. Here's another oxygen atom ion with a -2 charge. What's going to happen here? Look at it and tell me, common sense, what's going to happen? It's going to bind to hydrogen. Number 1 rule of chemistry, species don't like to be charged. They want to be neutral. Second rule of chemistry is these are kind of unofficial rules. Second rule of chemistry is you want a full octet. This satisfies both. If oxygen takes on the hydrogens, then what are you going to make? So you make water. In fact, you make 2 waters for every oxygen atom you breathe in. Oxygen molecules, sorry. So water is going to form and you form 2 waters for every oxygen that you breathe in. What did oxygen do to that last electron, as it accepted the electrons? What did it do to the thing that was in the chain? The last member. What did Jonathan do for Alana when he took her electron? What did he do to Alana? He oxidized her. She became more positive. Where Jonathan became reduced. So oxygen oxidizes other things because it makes it more positive. Because it took the electron, it becomes reduced. I know it's confusing. The terminology is tricky, and it's easy to get confused. This is one of the worrisome parts. This and cell signaling can be very confusing if you're not careful. But that's a real simple explanation of redox reactions. It's pretty, I think, accurate. If you want a more descriptive one, I can make it for you and print it out as a fact sheet. Okay? Now let's go ahead and look at citric acid. Yeah.
Kylie has a great mnemonic.
All right, go ahead.
It's called oil rig, right?
Oil what?
Oil rig.
Oil
Rig?
rig.
But you should write it like downwards. Like write it down like each letter. OIL in a straight line.
Oh, I see. I was going to do the opposite. I was going to write those words you're about to say in it. Okay, but yeah, okay, I can do that. OILRIG. Okay. What?
Oxygen
Oxygen
is
losing
losing,
electrons. Okay.
and then production is gaining.
Okay. Nice. And you've heard this before, Alex?
Yeah.
Excellent. Yeah. Organic chemistry, you do this a lot. This is organic chemistry. Everybody complains about organic chemistry that it's a. I loved organic chemistry. I would have been an organic chemist. I love it. I thought biochemistry drove me crazy while memorizing every word, every enzyme. Oh my God. So many pathways. But organic chemistry made sense to me. Follow the electron, right? That's all it was. Find the electron, follow it. It was almost like a puzzle, a game. But that's really good. Oil rig. I think I might have. I think I might remember that mnemonic from when I was in college, but I can't. I don't know. I don't remember it. I don't really use mnemonics. It's just for me, it seems like it's extra work to remember more words to remember. But it does work. I know this research shows it does work. So oil rig works, so that's good for you. All right. Are we good? Thank you for sharing, Kylie. All right. Easily drawn. Again, when I look at this, I know these are hydrophilic. I know these are hydrophobic. Says I have to put sodium ions on the outside or on one side. I'm not going to keep putting Na, so I just put a bunch of pluses. There's a bunch of pluses. And the question is, will something move through this membrane? If so, what? So we know for a fact the sodium ions will not move without facilitation, which means help. They need a protein. So they're not moving. That's correct. But if the ions don't move, what can move is water. So water can move. And water will move water will move from its high to its low, and we call this osmosis. How many people got it right? Excellent. In what direction did you draw the arrow? Huh? Which up, down, down, up? You do it like this? It's incorrect. Nope. Well, we'll go both ways, but water is going to move from what? Okay, from what?
High to low.
No. From what? You're not that you're wrong, but from what?
It's high to low.
It's high. So consider this side. This side, it's high in solute, correct? Yes? Everybody okay with that? So what is water? If it's high solute, it's low water. High salt. The salt is sodium. It's the solute. So high solute means low water, correct? All right, so to remind you, you have a pie chart, it's 100%. If I have high salt, there's a high salt concentration, I don't know, let's call it 30%, then what percent is the water? 70. Correct? On the other side, I have 0% salt, correct? Yes or no? Remembering that that's a solute. So what percent water is it? So where's water higher? On which side, up or down? So in this lower part, water's higher. So water's high here, and solute must be what? Low. Wherever solute's high, water's low. Wherever solute's low, water's high. And whatever's happening on one side of the membrane, the other side of the membrane's opposite. Opposite, opposite, opposite. So if you know this is where the salt is, you know that's high solute, that means the water's low. That means the other side of the membrane, salt must be low and water high. You don't have to do the pie charts. You just have to know that the opposite side is opposite. Are we clear? So solute high, that means water's low. Solute's low, that means water's high. You're right that if the salt could pass, it would move downward. It would go from salt's high to salt's low. But you're not worried about salt's concentration, are you? Are you? You're worried about water because water's what's moving. We call this osmosis. So you go from water's high, which is here, to water's low. So the direction of the arrow should have been up. It's a horrendously big arrow. Should be up. Correct? Yes or no? All right. The OST, they're going to ask you these questions. And as you saw in your final for the last semester, they'll give you cells. One's going to look small or shriveled. Another one will look normal. And just the example, this is just the example they gave. Another one's swollen. Then they'll give you an experiment where they'll say, well, look, here's this one. And this one's in 10% solution. What are the percents of each of these? Well, if this is a 10% solution natural, this is blood, whatever, then you'd have to say that this one is isotonic. So this one must be 10% because they're the same. They're not shriveled. It's not swollen. So it's 10%. This one must be higher than 10%. Why must it be higher than 10%? Because water's leaving. And water moves towards where it's hypertonic, where there's more salt and less water. So if it's leaving, it's shrinking. And here, this must be lower than 10%. And I say higher, it could be anywhere from 10.00001 all the way up to, 100%. Whatever. 10% is equal. These two have to be equal. And I don't know why I put low. I guess I'm thinking of lows. Low. Oh, maybe it was lower. Yeah, I was probably writing lower. Lower means anything less than 10%, so 9.999, whatever, all the way up, all the way down to 0% is fine. Any number would do. And that's going to swell up the cell. Why? Because water's going to go in. Why is water going in? Because the inside is hypertonic. The inside is high salt, low water. Outside is low salt, high water. Water moves from high to low. So that's osmosis. Again, you will get this again tomorrow and the next day and the next day and the next day until you get it right. Are we all clear on this? I'll post this 8-minute video. When I post this, it will have the redox reaction explanation. If you want to know what oxidation reduction means, you can look at it. I don't want to spend class time looking at it because I don't think you need to know it in that depth, but it does explain some things you might want to be interested in. What, like, for instance, why is this called oxidative phosphorylation? Cellular respiration occurs in 4 phases: glycolysis, the link reaction, Krebs cycle, and the oxidative phosphorylation. Just to be clear, oxidative phosphorylation, another word for it, is electron transport chain. Which phase of cellular respiration occurs in the cytoplasm? Everyone should be able to answer that right now. What is it? What happens in the cytoplasm? Glycolysis. Perfect. Which phases of cellular respiration occur in the mitochondria? Link reaction. Yeah, and yep, that's true. And Krebs. All right. Krebs and the electron transport chain all happen in the mitochondria. I'm going to label them 1, 2, 3, 4 steps in cellular respiration. Remembering fermentation is what happens when there's no oxygen. So all 4 of all of these answers here are right there. They put them in black highlighted or bold-faced type. I'm going to use ETC because it's easier than oxidative phosphorylation. I always put it phosphorylation, which I don't know why I do that, but whatever. Which of the 4 phases of cellular respiration require oxygen? Of these 4, which is the only one that requires oxygen directly? Huh? That's right. It is directly ETC, but indirectly it would be all 3 of these. So this one's the one that actually uses the oxygen. But if you don't have oxygen for this one, then the whole chain stops. Everything stops. And that's why when you have pyruvate, remember what goes into it, we'll go over that again. But as I showed you in the original diagram, you can see it in the original diagram, it's pyruvate, right? That thing I told you had to memorize. Pyruvate is what goes into the link reaction. If there's no oxygen gas here, then the pyruvate goes to fermentation. These 3 depend on oxygen. Now I want to say this while I'm recording because I think it's important that you understand this. Let's say that young lady back there is the end of our chain. So you got the link reactions right here in the beginning. In the middle, you got Krebs. And at the end there, you got the electron transport chain. And the last participant in the chain is that young lady back there. Look at her. She's all by herself. And over here, we got this giant pile of hot potatoes. The hot potatoes represent the high energy electrons. Okay? Are we clear? Mm-hmm. I mean, it's a giant pile. You just ate a candy bar. It's a giant pile of high energy electrons. I start passing it down. I bring it over, and I hand it off to the first person, the link reaction, and she hands it off to the next one, the next one, the next one, all the way down the line until she gets it. And I keep handing off these electrons. I keep handing them off. What happens if no one takes that last? You can only hold one of the hot potatoes at a time. Pay attention, because you can't. If you're not, leave him alone and focus on what I'm saying to you. What is going to happen when we all have hot potatoes and we can only hold one at a time, and nobody can take that potato from that last person in the chain? It's going to be backed up. It's over. The whole thing stops. All our hands are burned. We need somebody to come in from outside and grab that potato and leave so that the chain can keep going. Who's the last? What is that thing that comes and gets that last potato? Make a guess. No, pyruvates was bringing it into the chain. What's the last thing that picks up that
ETC?
from the ETC, has to take that electron and leave? What is that thing? What's that outsider called? Oxygen. Oxygen is the final electron acceptor. Without oxygen, this whole chain stops. From the link reaction to the electron transfer chain, the whole series stops. That's why we have fermentation. All right, have a good one.